Good afternoon, everyone, and thank you so much for joining us for this very exciting webinar. It's our first for the webinar series that will be running in the next few months. We don't know when it's going to end, but we're so excited about it. It will be focused on the value of natural science collections. Before I go into that, my name is Fulubero Tambani. I am the Science Communication Officer for the Natural Science Collections Facility. In short, we are the NSCF. If you look at the screen, we have our social media platforms that you can join us on. If you just use those handles, you'll find us very easily. Also, our website is www.nscf.org.za. Now, if you have no idea what the NSCF is, we are a network. So we are a network of South African institutions holding natural science collections, established as part of the Department of Science and Innovation's Research Infrastructure Roadmap. And we are also coordinated by the South African National Biodiversity Institute, which is SANBI. We have 16 partner institutions, and in these institutions are over 200 staff members, all these making up the NSCF network. I'm going to introduce the guests to you. We have 3 guests from some of these partner institutions, and they will be giving presentations about the work that they're doing, which has impact or which contributes to food security and agriculture. Firstly, we will have Erich van Wyk, who is from the South African National Biodiversity Institute, SANBI. He is the manager of the National Herbarium there. His work focuses on collections management, plant ecology, and conservation science. He has been in the field for 21 years. 11 of those, he has been in conservation science, and 10 of those has been in collections management. What Erich loves most about his work is seeing people in natural science collections, passion for and loving and nurturing the environment through their lives and in their workspaces. After that, after Erich will be Chantelle Girgan from the Agricultural Research Council, in short it's ARC, which most of you will know it by. She is the curator of the National Collection of Nematodes and a specialist on one of the most destructive nematode families, Pratylenchidae. I hope I pronounced that correctly. Chantelle was introduced to nematodes with the start of her honors degree. She fell in love with the nematode morphology while studying towards her master's degree. Now, during her PhD, she was introduced to the National Collection of Nematodes, and it was love at first sight. Apart from nematodes' importance in agriculture, Chantelle is fascinated by free-living nematodes and continues to study their diversity in South Africa. Chantelle has a passion for nematodes, and her favorite part by far is sitting behind a microscope, staring at these microscopic giants all day. Now, our last speaker is Riana Jacobs-Venter from ARC also. She is the curator of the National Collection of Fungi. Her research work focuses on fungal systematics, specifically on the toxigenic genus Fusarium. She has been in this field for 23 years. And what she loves most about her work is the diversity of fungi and their ability to adapt to challenges. What amazing speakers we have. I'm so excited. I can't wait to hear what they have to teach us, as most of you, I believe. Now, before we get started, we just need to go through some webinar guidelines of engagement. If you can please keep your contributions helpful and considerate of the host and other participants. So in short, please be kind to other people in this room. Use the chat box to say hello. Let us know which organization or school or university or whatever is applicable to you, where you're joining us from. You can do this right away. Use the chat box to say hello to everyone in the room and let us know where you're joining us from. You can also use the Q&A box to add questions during the webinar. I'm sure that as the webinar goes along, you'll have some questions. So you can just note those in the Q&A box and we'll get those answered at the end of the webinar. If time is not on our side, as it usually happens, we might not be able to answer all questions, but if that happens, we'll try by all means to answer those on the webinar link. This webinar will be posted on our YouTube channel, which is Natural Science Collections Facility. So if you go to YouTube and search for that, you will find it. And please remember to subscribe while you're at it. You can visit our website, www.nscf.org.za, to get to know us, our staff members, what we do, what we're all about, our partner institutions, some information about collections and their value, everything. You will find it right there. I see some people have greeted. We have Shahida Davids saying hello from SANBI. We have Karin from ARC. We have Geraldine, Isabel, Petro, Kim, Lawrence. Thank you, guys. Thank you. And they keep coming. Cameron, thank you for joining us. And please keep saying hello to those who haven't. Now, we are going to have an introduction to collections, natural science collections and their value by Chantelle Ribeiro. Chantelle is the project manager for the NSCF, and she'll just let us know what these collections are for those who have no idea and why are they important. Chantelle?
Thank you, Fulu. And thanks to everyone that made the time to join us today. Those of you who are part of the NSCF, and also welcome to our guests that are not part of the network. I'll be giving a quick overview on what natural science collections are all about and why they are so important for us as South Africans, but also for humanity as a whole. So the collections we are talking about are preserved plant, animal, and fossil specimens and their associated data, which are held by museums, science councils, and universities. So here are some photos that show how the collections are stored. These specimens are preserved in a variety of ways, be it the dried and pressed herbarium plant specimens, or the pickled, as we like to call it, wet specimens, which are preserved in alcohol. We also like to think of our collections as a collection of people. The staff that are employed in these collections look after the specimens in many different ways. Without the dedicated people behind the scenes, the collections will be lost to research. The staff also have a very important job of creating and maintaining the data associated with each specimen. Each specimen comes with a label and a corresponding record in an electronic database, which names and classifies the species, capturing the location where the specimen was collected, when it was collected, by whom, and so forth. And every specimen with good data provides a physical snapshot of a species or a community in a particular point in time and space. It is this physical scientific record that makes collections so valuable. The specimens tell a fascinating story of life on Earth, from the earliest origins to how life has evolved in the way that we know it today. The collections are a permanent record of existing and extinct animals that would otherwise be long forgotten, and as would our understanding of the intricacies of evolution. Then, as Fuller was explaining, the Natural Science Collections Facility is made up of 16 of these institutions countrywide who are the custodians of the collections. We are a network of partners committed to building a natural science research infrastructure for South Africa by making sure these collections and data are safe and properly looked after according to international best practice and standards. Unfortunately, natural science institutions in South Africa are often underfunded, understaffed, and undervalued. But they are valuable beyond measure, and the Value of Collections project aims to showcase this value. Collections are important for a number of reasons. They are built up over 200 years, and they present economic and scientific opportunities and are used by researchers all over the world. This includes research into agriculture and fisheries, human health, enforcement in the illegal wildlife trade, climate change adaptation, and indigenous knowledge systems. Many, if not all, of the uses of collections have an impact on societal issues that afflict the contemporary world. These do not only affect global issues, but also local issues that affect people and communities directly. So this webinar and the linked showcase focuses on how the collections contribute to South Africa's food security and agricultural needs through pest and disease control, decisions about imports and exports, predicting viability of crops. And the PDF version of this showcase or this webinar is published on our website. It was released in July, and up at the top of your screen, you will find a link where you can go and read more about the research that's being presented today. Thank you. I'll hand over back to Fuller.
Thank you, Chantelle. As Chantelle said, the showcase is on the website, and just so you know, the showcase is linked to this webinar. So the authors of that showcase are the presenters today. We can now get started with our first presentation or talk. It's by Erich van Wyk from SANBI, who will present on Crop Wild Relatives project. So on the showcase, he actually co-authored the paper on, or the article on the showcase with Chantelle, who was just, and they titled the article "Family Ties and Safety Nets." What an interesting one. Let's hear from Erich.
Thank you for the opportunity to speak to you today. As part of the theme of value of collections, I would like to tell you how our herbarium collections and the very important information we get from the specimens contribute to food security. I want to start this presentation by talking about food security. As you know, and can tell from this picture, the term might mean different things to different people. The United Nations Committee on World Food Security, though, define it by saying that people should at all times have access to sufficient, safe, and nutritious food that meets their needs for an active and a healthy life. The harsh reality is that although there is estimated to be more than 7,000 species that has been used for food, most of today's food is derived from only 30 species. Let that sink in for a moment. To go even further, it is estimated that over 50,000 plant species in the world might be edible. It is staggering to find that only 10 crop species account for 60% of the world's caloric food intake. These include grains, a few tuberous plants, some legumes, yams, and a member of the banana family called plantain. Amazingly though, more than 50% of the world's population relies on the top 3 grains alone. No surprise, these are maize, rice, and wheat. The really bad news though is that our food security is under severe threat. Over the coming decades, a changing climate and other environmental stresses will have a significant impact on food security. In some parts of the world, this will result in reduced crop yields from heat stress and reduced water availability. On the other hand, it will lead to extreme weather conditions, and large storms are likely to cause flooding that will have disastrous effects on crops in the region. The crops will literally drown. As if too wet or too dry crops is not bad enough, too wet or too dry conditions will also lead to other problems like disease and pest outbreaks. And this will eventually lead to even more harvest losses. Okay, that was the bad news. Now for the good news. We are already doing something about this. The good news is that we can change crops to be better adapted to the pressures of climate change. To be able to do this, it is essential that we understand, that we conserve our crop world relative plants. Wait, what is crop world relatives? If this is a new word to you, these plants are wild, or if you like, naturally occurring plant species that are closely related to cultivated crops. These species can potentially donate beneficial genes to crops in breeding and plant improvement programs. To put it very simply, crossbreeding a cultivated crop with a crop wild relative with beneficial genes can lead to a plant better adapted to a different environment. One example is a plant used to a cool, wet climate could, through several iterations of crossbreeding, be better adapted to a warm, dry environment. I'm no expert in plant breeding, so there's of course a lot more detail to this. Firstly, I said that we need to understand crop wild relatives better. A part of that is understanding the taxonomy of these species. This is key to unlocking their potential in plant breeding programs. Our taxonomy is based on studying our plant collections and understanding that taxonomy and classification of plants help to identify plants with potential traits, and these traits will include things like improving tolerance to abiotic pressures such as drought, heat, and flooding, improving crops by increasing their resistance to certain pests, and in general, overall increasing of crop yields due to better adaptations to the new environments. Taxonomy gives us the basic understanding to be able to identify the resistance and adaptive traits necessary to provide crop stability in a rapid changing environment. It is very clear that conservation of crop wild relative species is vital for ensuring future of food security. And our collections provide some of the basic ingredients to achieve this. Information associated with plant specimens in science collections is useful in developing conservation plans to ensure future protection and survival of crop wild relatives. We use collections, for example, to develop a list of all South African plants, and we call that the South African Plant Species Checklist. Specimen data of specimens in science collections provide us with distribution data for plants and also their level of endemism, in other words, to which area or region they are limited to. Specimen data also often contains information of plant use, and it also forms the basis for threat assessments and many other examples. The South African development community recently participated in a worldwide project on crop wild relatives. In South Africa, SANBI partnered with the Department of Agriculture, then it was called the Department of Agriculture, Forestry and Fisheries, and the ARC in running this project. SANBI Iberia was tasked with producing a checklist of wild plant relatives of human food and fodder plants. What we did is we compared a list of known crops with our South African plant species checklist and identified relatives at the genus level. This resulted in a crop wild relative checklist of 1,593 taxa. That's almost 7% of the country's flora or taxa. That's quite a lot. So the list was therefore further refined through a prioritization process which considered the following: socio-economic value of the related crop, the potential use of the wild relative in crop improvement. Obviously, you don't want to cross or breed a poisonous plant with a crop, hoping to achieve anything. The conservation status, and then whether it is restricted to South Africa, or in other words, an endemic plant. Now, most of this information used originated at some level or another from specimen data. This prioritization process reduced the original list of 1,593 taxa to 258 taxa belonging to 33 genera and 15 families. 93 of the 258 taxa on the list were endemic to South Africa. I know it's difficult to see from the graph, but the predominant families on the list was the Poaceae, that includes several grains, the Fabaceae, which are the beans and the peas, and the Solanaceae, the potatoes and sweet potatoes. So all available specimen records for these 258 crop wild relative taxa was gathered. It was collected from different herbaria and citizen science virtual museum platforms, and it resulted in a large data set of about 23,527 specimen records. Now you can see on the map the distribution of these taxa, and you can tell that it covers basically the whole country. After a process of data cleaning and georeferencing the data, it was provided to a conservation biologist to conduct a spatial biodiversity planning analysis. To very briefly explain, they use systematic conservation planning methodology to identify the best sites to conserve priority crop wild relatives, and at the same time they determined which species are already protected in formerly protected areas. They then prioritized new conservation areas based on the following: the level of endemism, the level of endemic crop wild relatives protected in this area, and also then the overall species richness of the area in South Africa. Again, there's of course a lot more detail to this process that I have time to share with you in this presentation. So the most important sites for protecting crop wild relatives in South Africa was therefore finally identified. And finally, the last step was to engage with conservation authorities and stewardship programs to ensure that these sites are prioritized for inclusion in future protected area expansion strategies. This information was also included in the National Strategic Action Plan, and there it sets out a coordinated, systematic, and integrated approach to the in situ and the ex situ conservation of our crop wild relative diversity. I want to thank you, and that's all from me. I really hope that you've enjoyed the exciting virtual tour of our valuable science collections for conserving crop wild relatives and enhancing our food security. I thank you. Back to you, Fulu.
Thank you. Thank you so much, Erich. Well, I know I don't want to go hungry, so I'm very grateful for this project. It's so good to know that there's backup for food security. So thank you so much to you and your colleagues for all of this work. Now we can move on to our next speaker, Chantelle Girgan from ARC, to talk to us about the National Collection of Nematodes, those microscopic giants as she describes them. Chantelle?
Okay, good afternoon, everyone. Thank you, Fulu and Chanel, for the opportunity. Today, I want to tell you about the National Collection of Nematodes, what we do, and the role we play in food security. First, I'm going to tell you what a nematode is, as some of you might not know. So, nematodes are from the phylum Nematoda. They are unsegmented worm-like organisms, and their size ranges from 80 micrometers to 8 meters. Now, I know 8 meters is not microscopic, but the nematodes we work with range from about 80 micrometers to 4 millimeters. The 8-meter nematode is one that's actually from a sperm whale, the parasite from a sperm whale, and it's also 2.5 centimeters in diameter, so it's massive. But we don't work with anything nearly as big. Nematodes are also extremely abundant and diverse. The father of nematology, Professor Nathan Cobb, once said that if you had to remove all the trees and the rivers and the mountains and the people from Earth, you would still see the outline of everything in the form of nematodes. So that gives you an idea of how many nematodes there are. They're also found in all environments and on all the continents. And the current species estimate is about 1 million species, but sadly only 97% of that is unknown. So we really do have our work cut out for us. So nematodes are divided into two groups. You have the free living group, which is terrestrial and aquatic forms. Then you also have the parasitic forms, parasites of plants and animals. So to tell you a little bit about our collection, we have one working collection and two donated collections. The National Collection of Nematodes was established in 1955 by Professor John Haynes, which is the father of nematology in South Africa. Our collection currently has a little over 50,000 slides with more than 200,000 specimens of 314 genera and just under 1,400 species. Now, we are very lucky to have almost 7,000 type specimens in our collection, and these are holotypes and paratypes. All our specimens are also connected to specimen and locality catalog cards, which are handwritten, if you see here in the middle. And they are also linked to a very extensive literature collection, which mostly includes also the original descriptions of the specimens in the collection. Then the second collection we have was donated by Dr. Kent Kleinans. It's the Molodagani collection, or the root knot nematode collection. It was started by Dr. Kent Kleinans as a survey of South Africa of the genus. There's about almost 15,000 slide mounted specimens. And if you remember my previous talk at one of the NSCF webinars, Molodagani acronia was the first plant parasitic nematode described from South Africa. There's a female in the picture over here. And very recently we found its life cycle in a box hidden in one of the cabinets, which is about 60-year-old slides, which is now fully incorporated into the collection as well. Then we also have a third collection, the John Haynes collection, which was donated by the late Prof. John Haynes in 1999. There's about 7,300 slides, which consist of 21,900 specimens of plants as well as free living and aquatic nematodes. And this is Professor John Haynes previously mentioned. So now to come to the value of our collection, or the uniqueness of our collection. The National Collection of Nematodes is one of the largest nematode collections globally. The USDA in America has little over 200,000 specimens, and we have 200,000 specimens. So we are like right up there with them. We're also the biggest, if not the only, reference collection in Africa. We record history as all collections do, and our oldest holotype was deposited in 1957. We also now, for quite some time, fully digitized in two databases, the National Collection of Nematodes database and the South African Plant Parasitic Nematode Survey database, and the two are connected. We also do training of students, and our data is available to anyone who asks for it, scientific or citizen scientists. Then another really thing that I see as a value of our collection is our taxonomic expertise. I would like to introduce you to our team. We are a team of ladies. We have Dr. Esther van den Berg. She is a retired taxonomist. She's been retired for nearly 16 years, I think. Then we have Dr. Antoinette Swart. She's also a specialist working with us. Dr. Mariette Mare, another specialist, and then me, and then Mrs. Adoration Shubani and Mavis Haji. They are our technical staff, and without this team, the collection would be at a standstill. So the value of the collection is also the services we render. So we do diagnostic services daily, and annually we identify over 2 million specimens and from about 250 something plant species. We also do soil health assessments, and all this contributes to food security, and I will tell you why. So whenever you hear about nematodes, you probably hear about the plant parasitic nematodes. According to the American Society of Phytopathology, 14% yield losses in the agricultural sector globally is due to plant parasitic nematodes. Now this equals to about $125 billion annually. So the identifying of these plant parasitic nematodes is crucial to prevent these yield losses, because if you identify a nematode correctly, you can manage it correctly and prevent these yield losses, which contributes to food security. So plant parasitic nematodes might be tiny, but I call them microscopic giants for the following reason. They are literally micrometers in size, and they cause extensive damage to crops. Here you can see some of our quality crops that we grow in South Africa, and they don't look so delicious because of nematodes. So plant parasitic nematodes can cause total crop loss, poor quality of crops, and unfortunately they have an extensive host range, which makes identifying them crucial. And also here comes the value of a collection. The distribution of them is crucial too, because if you know where they are and what their host range is, you can correctly manage them and contribute to food security. So one of the databases we have is the South African Plant Parasitic Nematode Survey database. It's a mouthful. It was founded in 1987, and the goal was to create a comprehensive assessment of the nematode biodiversity resources of South Africa. Now, as you can see in this map, we have more than 8,500 locality points. Each of these points has the information on the plant parasitic nematodes and more recently free-living nematodes, host range, where it was collected, under what circumstances it was collected. So it's quite an extensive database, and it directly contributes to food security, because as I previously said, you need to know what, where, and how you have it to be able to control it. Now to talk about the good nematodes, the free-living nematodes. And these little creatures also contribute to food security because they play an essential role in the soil, especially in nutrient cycling and mineralization. They are also used as biological indicators of soil health. They're also used in biological control of other pests. These are called entomopathogenic nematodes. Now, this graph is one of the tools we use to establish your soil health based on the nematode communities. I'm not going to go into a lot of detail, but it's divided into 4 sectors. And depending on the free-living nematodes you have in your soil, we can plot your soil in one of these sectors and tell you what the status of your soil health is. So some of the examples of the work we do, highlighting the value of the collections in food security, is some of the projects that I've listed here. So firstly, we have the Potato SA project that studies nematode communities in crop rotations in a potato field. We also have SAMAC, which is the Macadamia Association, where we study the nematode communities in macadamia. And previously, there was very little information on the nematode pest species of macadamia. We also do nematode diversity of natural grasslands. My PhD was in Tapira Nature Reserve. And this is important because a lot of nature reserves are unfortunately used for agriculture on a later stage. So if you know what you have there, you know the potential pests you might have. Then we also have a very nice highlight. The data of the collection was used in a Nature article. And this is a global, I see a spelling mistake, in a global scale. So this data, with data from all around the world, was used to show the distribution and spread of nematodes in the world. We also have had a master student, or one of our technical staff did a master's on the characterization of Heterotera carotae, which is a very big pest species on carrots. And then, of course, we do work for the large on quarantine and phytosanitary. So I really hope that I highlighted the important role that we play in food security. And if you have any questions about our collection, what we do, or would like to be part of it, you're more than welcome to contact me. Thank you.
Thank you so much, Chantelle. Microscopic giants indeed. I'm sure that if we have any farmers in the house, or even those that will watch the recording, they'll be very appreciative of all this information, even those that are planning to go into farming. So thanks so much, Chantelle. We will now go on to our last speaker, Riana Jacobs-Venter from ARC also. She will speak on the National Collection of Fungi. So she asks a question: growing in the dark, fool or friend? Now let's hear from her what she says. Riana?
Thank you very much, Fulu, and thank you very much to the NSCF for this opportunity to share my passion about things that grows in the dark. A leading group of mycologists wrote a paper a year ago on the commercial uses of fungi in the world. And they listed up to 50 different uses, including the production of biofuel, a food source, environmental management, in which we've used the fungi to remediate contaminated soils, in agriculture, and of animal feed, to name but a few. But the most important role, or the most well-known role of fungi, are those of plant pathogens, where they affect forest crops like the pitch canker outbreak we had in the country. It impacts on banana production, tomato production, and many other vegetables and fruits, and of course, flowering plants and cereals. An important component of the infection of fungi on cereals are toxin production. And these toxins can actually stay throughout the entire production chain and end up in the food on our shelves. So in South Africa, the history of mycology and plant pathology dates back to 1905, when the collection was started by Paul Evans under the mandate of the British government. And for the past 116 years, it has grown to be the only and obviously the biggest collection in Africa. But there are two important role players that we need to acknowledge in this journey of 116 years. The first being Ethel Mary Deutsch. Mary was the first lady to obtain her PhD, although not on fungi, but on bacterial diseases. And she, in 1945, published the first document on all fungi associated with agricultural crops in the country. This was updated in 2000 by Professor Dr. Pedro Kraus, and publishing a book on the updated list of plant pathogenic fungi. But Pedro is also known for his huge contribution to our knowledge of fungal species occurring on fynbos, and he has described up to 100 species, now being the director of the biggest fungal collection in the world, the Westerdijk Fungal Biodiversity in the Netherlands. So the National Collection of Fungi consists out of two different collections, Pre for Pretoria and M for Mycology. And this collection was, as I said previously, established 116 years back. It accommodates 63,000 dried specimens, which represents over 12,000 species and 2,300 types. It also incorporates an extensive literature reference library that dates back to 1755. It includes both South African and foreign specimens from all the continents, but a significant part of this collection was established by Ethel Deutsch, about 40% of this collection, through her collaboration with African and European mycologists and exchange of material. And we have a significant number of specimens that presents African fungal biodiversity and not only South African biodiversity. And currently, our main purpose is to provide these material on loans to international and national researchers and to serve as a depository for type material. The second part of, or the second section of the National Collection of Fungi is the PPRI collection. It accommodates 29,000 living isolates, and these are preserved either in a public collection or a reserved collection under the Budapest Treaty, or the CBD and the Nagoya Protocol. And of course, we then request the PICs and the sampling permits to be in place for those different collections. Currently, these collections are expanded by our own research that is conducted in the unit. So other South African researchers that deposit their material in the collection, commercial companies depositing their strains that they use for commercial product development under the Budapest Treaty, our diagnostic services to commercial farmers, and the public that brings in most interesting fungi. An example of the value of this collection is a study that was just published in January this year on wheat brown rust. They used material deposited in the collection between 1906 and 2010 to study the genetic development of the populations of the specific pathogen of wheat. They found 5 subpopulations that survived between 21 and 82 years in the field. 3 of these representing entries from the European settlements. 2 appeared as exotic introductions, probably from other African countries. And clearly illustrating how important surveys are, but also the preservation of the results of these surveys, so that people can go back and study these and trace back the development and the resistance built up in these pathogens. Fungi are also used in agriculture in a beneficial way by production of biofertilizers, and these are mostly the mycorrhizal fungi, those that live in close association with their plant host, and you only see a very small part of this organism's life cycle by the mushrooms that are produced annually around the trees. And again, we host these specimens for registration. An example of these mycorrhizal fungi being used in the country is Mycoroot, which are a South African-based company producing mycorrhizal-based fertilizer. Fungi are also used in biocontrol, and an example of this is the commercial product that BASF has on the market called Broadband, that's based on material from the collections. Not only would they, this specific product is against insect pests, but we also have fungi that control nematodes, weeds, and other fungi. Then our diagnostic services, we are a DLARD registered laboratory, and then thus we certify material for import and export. The two fungi we concentrate on here are Tilatea indica, or coral bunt of wheat, in which we certify consignments clean of this organism before it's exported, and Osteolaga maydis, although it is seen as a delicacy in many parts of South America, in our country it is seen as a quarantine pest. And of course, we analyze soil, seed, and other plant material for commercial farmers and agricultural businesses. An example of the veterinary importance of these collections is the Kikuyu poisoning of cattle. Previously, it was reported that Fusarium fluorosum is the causal agent of Kikuyu poisoning in cattle. However, we found by means of comparing the South African isolates that we obtained from the Eastern Cape with those strains in the collection, that another species, where Fusarium species, were involved in Kikuyu poisoning in South Africa. And an important information that we need to distribute to the vets and the cattle farmers in the country. So the take-home message from the National Collection of Fungi is the agricultural industry contributes an enormous amount or a large portion of the gross domestic product. It contributes to food security, social welfare and job creation, and even ecotourism. Thus, it's important to support this industry, and that's why the collections play a vital role in the support system. And the NSCF has contributed this in past 116 years, and we would like to continue our contribution to the agricultural industry in future. Thank you.
Thank you so much, Riana. I'm a big fan of mushrooms, but I also know that they can kill me. So they are very interesting fungi, are very interesting organisms. So thank you for such amazing presentations by each and every one of our guests. Thank you for your time and putting in the effort to prepare and to come and give us this valuable information. I'm sure you can hear from all these presentations that collections, natural science collections, are very important, and they are very valuable, and their value varies across disciplines. So I hope you have learned something new from this webinar. On that note, I think people are satisfied, and I believe we can now close. Thank you very much to everyone for taking your time to attend this webinar. It was our very first on the webinar series, and we are so glad that you joined and that you found it valuable. Look out for the next one in August, where we'll focus on the theme pests and diseases. We'll start with publishing the showcase about it and then follow by the webinar. You can keep in touch with us on our social media, on Facebook, Twitter, Instagram, on YouTube, where we'll post the recording of this webinar, and you can go there and also direct your colleagues and friends to watch it, as well as on our website, nscf.org.za. And you're welcome to leave your comments or suggestions on the link, which will be on YouTube. There will also be a recording on Facebook, on our Facebook page, since we are also streaming to there. So that's it, everyone. Thank you so much. Thank you to our speakers, and thank you to everyone who attended and for contributing to this webinar. We appreciate you. We are grateful. And thank you again also to Chanel for being our technical help. Thanks so much. We'll see you at our next one.